Hello, everyone. My name is Angeles, and I'm a PhD candidate at the Conesa Lab in Valencia, Spain. And today I'm going to do part of what was originally conceived as a long-read tutorial. And I'm going to cover the basics of what functional isotranscriptomics analysis with TappAS is. And so this is going to be a very practical presentation, very focused on how to use the application and how to get around the interface and the possibilities of analysis using TappAS. To better understand the aim of TappAS, I'll first explain what functional isotranscriptomics analysis is. Traditional isoform expression analysis evaluates differential expression of the different isoforms of a gene between conditions, and it can provide information about changes in the major isoform that is expressed in each of them. However, this approach presents several limitations. First, it's not very easy to systematically visualize the differences between isoforms that have been generated due to splicing. And in addition, the biological readout of these changes in expression is very difficult to obtain because there's no way to know how much the isoforms of a gene really differ in biological function. To make these two tasks easier for users, we have designed strategies to incorporate functional elements into transcriptome annotations using iso-annote and iso-annote. Iso-annote can actually be run directly with scanty3, which is something that my colleague Fran is going to cover in his own talk. But once we have these functional elements annotated at the isoform level, we provide in TappAS analysis that combine expression with these functional annotations to enable what we call functional isotranscriptomics. Furthermore, using TappAS interactive maps, such as this one here, we can visualize these features within the sequence in order to understand the functional differences between each of the isoforms. And before I dive into TappAS functionalities, I'll show you a brief example of how this works. So thanks to the functional notation that I have just described, we know the correspondence between these isoforms and their expression and the functional features, motifs, domains, and so on that they contain, both at the transcript and at the protein level. Take, for instance, this nuclear localization signal here, which is included in two of the isoforms in the catenin delta gene and is excluded in the other two. And by combining the expression of the feature including and feature excluding isoforms, TappAS can detect a change in feature inclusion that is condition dependent, with NLS containing isoforms being actually favored in the oligodendrocyte condition and the exclusion being favored in the neural condition, right? This is a neural development series that we've actually used in the TappAS paper. And this was an example of a splicing-based functional regulation that was validated at the experimental level in the publication. So although different approaches are implemented for different types of features, this very same logic is what we actually use in most of TappAS analysis, and it's at the core of the FIT framework. And so once we have these notions of what TappAS is actually for, I'm going to move on to describing how the application, the proper software, actually works. I'm going to give you a brief description of what TappAS inputs are, right? What files do you need to get TappAS to work? A quick guide to the application's interface, and then I'm going to move on to describe the different analysis options that are available in TappAS. So first of all, how do I create a project in TappAS, and what is a TappAS project? Projects are the main instance in which TappAS is based. A TappAS project is a self-contained workspace in which you will work on a single experiment using all of the data that's available within TappAS, storing analysis results, and independently doing whatever you want without affecting any of the other analysis that you have going on and that you're working on with TappAS. Project creation requires three main inputs. First, a design file, an isoform level expression matrix, and finally, a functional notation GFF3 file that can either be selected from those that are built in TappAS or provided by the user. So what is a design file? The design file is a small text file. I have an example here that specifies the names of the different samples and the experimental conditions that they correspond to. In the case of this tutorial, for instance, we have neural precursors, this here labeled as NSC, and oligodendrocytes, labeled as OLD. Note that TappAS is always going to require at least 2 replicates per experimental group, and is going to make, it's going to need for you to match the sample names in the design file with the column names in the input isoform expression matrix. It's very important to remember that isoform IDs do not require that a column name is provided. Only the sample names are accepted. And TappAS will make sure to make these the row names to your matrix internally. However, this case in which we have 2 groups to compare is really not the only experimental design that is accepted by TappAS. Other experimental designs are single time course, containing a single experimental group, but observations across several time points. Then we need 2 replicates per time point, and multiple time course, in which we have both multiple experimental groups and multiple time points. And again, 2 replicates per time point and per group. In this case, when you have time course designs, you need to provide the time column as well between the sample and the group columns. And finally, to create a project, TappAS requires a functional annotation file in a compatible GFF3 format. It's important to remember that TappAS GFF3 format is not the same as the regular GFF3 formats that you have available in your reference databases. We have a GFF3-like format in which we include both transcript, exon, gene annotations, and also functional features such as you can see here, motifs, microRNA binding sites, and some other type of gene level information such as gene ontology. And well, potentially you could add any functional feature that you were interested in. And TappAS, as long as it's in this format, and TappAS will be able to read this into the application. A similar file to this one can be generated using the --iso-annotate argument from Scanty. And well, Fran's going to give you more details as to how to do that. But if you're interested, you can always contact any of the maintainers and developers, and we'll assist with the analysis of your own data and help you obtain your own annotation. So after starting the TappAS application, you'll be able to supply the input files and create a project. I'm going to quickly give you an overview of how this is done. So you would open your application, click on the new project button here on the left, and a sub window will pop up for you to provide all the information for that project. In this window, you'll need to provide the following information, starting with a name for the project. Then you need to select a functional annotation. This can either be built in, right? You can use this menu here to select the species and the annotation, or specify your own path to an annotation file that is compatible with TappAS. In the next section, we're going to tell TappAS the type of experimental design that we're working with. And in this case, for the tutorial, we were working with a 2-group comparison, but if you open up this menu, you will have the 3 main options that TappAS can give you for your analysis. Then we need to supply a path to the design file and a path to the isoform expression matrix. And to correctly do these things, you have a help button down here that will tell you a little bit more about the formatting of the different files and also how to best choose your options for your data analysis. And finally, down here, we have 2 additional options. One is for normalization of the isoform counts. We use the TMM method, but if you want to do your own normalization, you can always unclick this and just add your normalized count matrix. And you can also apply an expression value filter, which will remove isoforms below a given expression level from your matrix. Finally, users can also provide an inclusion or exclusion list, which will subset the original matrix annotation files for your new project. Imagine that you just want to work on a subset of isoforms of interest. You just need to provide a single column text file providing the identifiers, and TappAS will just filter for you to just have this subset as a project. We very often get questions as to how to obtain some of these inputs, particularly the isoform expression matrix and the annotation file. And I just wanted to very quickly give you an overview of how we work with differential isoform expression when it comes to long reads. So generally, we can imagine that we have a two-condition experiment, like the one I was talking about before, right? And let's imagine that I have the same conditions that I was showing before, the neural stem cells and the oligodendrocytes. And to obtain long reads, I could just pull cDNA from all samples from the same condition, right? And sequence using, for instance, PacBio. And to complete the information provided by PacBio, some people like to generate short reads at the sample level. Of course, this varies depending on your design. You might want one PacBio run per sample or whatever. I'm talking at the condition level because what we normally do for analysis is we pull all of the long read samples prior to processing. And that way we can input the whole thing into Iso-Seq 3 and Scanty, and finally obtain a single GFF3 and GTF file containing the transcriptome and the functional annotations if you use iso-annotate. And this will be our experiment's transcriptome. For quantification, what we normally do is use the short reads from each of the individual samples and just map onto the reference GTF. And that's how we get an isoform level expression matrix. Of course, this workflow is going to be variable depending on what you're interested in. And there's a huge potential for direct long read quantification of samples. So the only important thing to remember is that you need one annotation per experiment because you need one file per project, right? So you can't really have one GTF file per sample when it comes to analyzing on TappAS. So once we have cleared this up, I'm gonna give you a brief tour of the TappAS application and the interface to get you acquainted with the software. After you create a project, assuming that everything runs smoothly and that you successfully created everything, TappAS will show you its main window. It looks like this, and it consists in three main areas. Up here, we have the control panel at the upper bar of the window. The data panel is at the top. You will see tables display here, other results too. And down here, there's the visualization panel, right? So you always have your analysis options up here, your tables in the middle, and your visualization, your plots, and everything that TappAS generates for you at the bottom panel, which is for visualization. The control panel contains three main areas. First, the projects button, which will provide access to the projects that already exist and give you the option to create a new one or delete one that's already there. Then a series of buttons, each of which contains several analyses associated to each of TappAS analysis modules, which I will describe later on. And using these buttons, users can open the results from whatever they have already run or run a new analysis. And a really cool feature of TappAS is that you sometimes can run the same analysis using different parameters and then compare what you got from each of them. And finally, here on the right-hand side, we have a search bar to filter the data tables that you are observing. You can insert a gene ID or a transcript ID, and this will be subset for you. When you select to run an analysis, you'll see a sub-window, such as this one. It's called the analysis parameter window. And in this window, we need to specify the analysis-specific options for a given, again, analysis. Whatever is displayed here will vary depending on what analysis you wanna run. Here I have the differential feature inclusion, but you can just explore the different options and use the help buttons at the bottom here, sorry, to just find more information about the parameters, what they mean for each of the analysis. Now for the data panel, it is designed to show tables, either the expression matrices for different features or results tables for different analysis, and all of this can be opened simultaneously on different subtabs. And remember that TappAS can open up to 3 projects simultaneously, and you'll see them here as a new main tab. This data panel has a multi-option sidebar, and this will give you some options to view analysis logs, filter the tables, export them, visualization options, and different things that you can do to tweak what you're seeing here. Using this plus button on the right, you can also customize the table, normally add new columns, but also remove them. And you can change these however you want and export the table entirely. And finally, here on the visualization panel, which you can open by clicking on the analysis graphics button, you'll see this window where you have different types of graphics. When you're opening the gene level visualization, you'll see gene specific options to view expression charts or annotations. And when you are playing around with analysis, you might see panels including some results plots or different things TappAS can generate for you on high quality and paper ready, just for you to export. Just remember, whenever you're playing around with TappAS, that there are multiple built-in documentation access points where you can just be like, okay, I don't know what this table exactly is. I don't know how to interpret what's in here. I'm just gonna go click on the little question mark and see what's in there. And it's sort of a learn while playing with the application philosophy. Finally, I'm going to really quickly describe TappAS analysis modules. Our tool includes many types of analysis, but this can be divided into 3 different modules depending on the aims of the analysis. First, there's the functional diversity module, which compares all of the isoforms for each gene and provides qualitative evaluations of the diversity of the functional labels in the transcriptome. This is like a tool to assess the level of variation that is potentially generated in the transcriptome, but this does not account for expression. For that, we have the differential analysis module, which includes both differential expression and differential isoform usage analysis, and it will detect changes in expression of each of the features that TappAS considers or changes in usage of isoforms within a given gene. This can be run considering transcripts, genes, or predicted proteins, and it also can be very easily coupled with different types of functional enrichment to give you a readout of which functional categories are actually enriched in those subsets of genes and isoforms. Finally, when you combine, this is the pretty much more fit type of analysis, it's the feature module, and it combines the isoform annotations with the expression as estimates that you provided. And so it can give you rates of inclusion or usage of different functional elements due to splicing. And we have here different types of annotated features or specific analysis for polyannulation and for global UTR shortening and lengthening analysis. Since time is limited in the presentation, I'm not going to say much more. If you're interested in using TappAS, you can go to our GitHub site and you will be able to download it there. There's more info and documentation both on the GitHub page and on the proper website for TappAS. And if you are interested in seeing how you can use TappAS to generate interesting results, how to interpret them, or how to get some ideas, there's a paper that was published last year where we demonstrate fully the application, and you can also go into the methods and really understand how the analyses are performed. And that is all. Thank you very much for your attention, and thank you to Liz for giving me the opportunity to speak today.